Welcome, everyone. Thank you for joining us today. Come on in. So welcome. This is the UMass Boston Dana-Farber Harvard Cancer Center Partnership, U54 Partnerships Annual Cancer Symposium. It's great to be in person again. And welcome here to our event. For those of you who are in Zoom, welcome as well. I'm going to hand it on over to my colleague, Dr. Abel, who will introduce today's speaker.
Thank you, Don. It is my great pleasure to welcome Dr. Alejandro Sweet-Cordero, who is a physician scientist focused on cancer biology and cancer genomics. He runs a laboratory whose goal is to understand the molecular rewiring that occurs in cancer in order to deepen our fundamental understanding of this disease and potentially identify new therapeutic opportunities. His lab uses functional genomics applied to mouse and human systems to understand signaling and transcriptional networks that regulate the outcomes of specific oncogenic mutations. And they have two primary disease interests, lung cancer and pediatric sarcomas. In addition to directing his own research laboratory, Dr. Sweet-Cordero also directs the Molecular Oncology Initiative at UCSF, an effort to advance the clinical application of precision medicine and oncology for adult and pediatric cancer patients. In June of 2022, he was appointed as the chief of the Division of Pediatric Oncology at UCSF. And we are thrilled to have him give us the keynote address for today's symposium, Genomic Analysis of Osteosarcoma, Making Sense of a Complex Genome to Advance Translational Research and Targeted Therapy. Thank you so much.
Thank you, Dr. Abel, for the introduction, and thank you to my good friend, Dr. Carmona, and also to Dr. Abel and all the other members of the partnership for the invitation to be here today and share my research with you. And thank you all for coming, and thank you to those who are on the Zoom. So I know this is maybe a topic that is not so familiar to many of you. So if I'm not being clear, or if you have any questions, please feel free to raise your hand, and I can try to answer any questions. And we can also answer more questions at the end. It's a real pleasure to be here also because I did my training here now over 17 years ago since I left Boston. So it's always great to come back and meet old friends. Keep trying to get this to advance here. There we go. Okay. So just the quick introduction. So my lab works on cancer biology. We're interested in several different topics related to pathogenesis of primarily lung cancer and sarcomas, and these are kind of the main areas that we work on. Functional genomics of oncogenic KRAS, tumor heterogeneity and therapy response, and then the pathogenesis of relatively rare but important for us as a pediatric oncologist, pediatric sarcomas. I'm going to focus today on our work related to the pathogenesis of one particular sarcoma called Ewing sarcoma. But just as a background, I just wanted to mention a few key facts that I think are important to keep in mind. Is that, despite the many advances that we've had in cancer research and cancer treatment, most patients, at least pediatric patients with metastatic disease, still remain incurable and will die of the disease. And that's partly because we don't really, even to this day, understand really what are the mechanisms that allow metastatic spread, but more importantly, perhaps what allows cells to survive in the metastatic niche. So once the cell gets to a metastatic site, why does it survive and not respond to chemotherapy? And because of that, we also have a limited ability to define which patients will metastasize and to tailor the therapy to prevent relapse. And in many pediatric solid tumors, targeted therapy, despite the advances we've had in a lot of adult cancers, still has had a limited impact. And many of these patients are still treated with chemotherapy that we've been using for many years. And again, despite the advances in immunotherapy, which has been quite exciting and really a transformative area in many solid tumors as well as leukemias, that has had so far a limited impact in pediatric solid tumors. So today I'm going to talk to you a little bit about the landscape of the genome of a particular cancer that's of interest to our lab called osteosarcoma, which is a rare disease, but I think because of the genomic features, this complex genome that I'm going to tell you about, the lessons that we could learn from this disease could be applicable to a lot of other pediatric cancers and adult cancers as well, where genomic complexity is a key feature. Then I'm going to move on to talk a little bit about the epigenome of osteosarcoma, which I think is equally interesting. And then finally talk about some approaches to how we're using this information to try to drive the development of new therapies. So osteosarcoma, as the name implies, is a disease of the bone that can really occur in any disease in any bone in the body. Most commonly seen in the long bones, that's shown here, but really can happen anywhere. It's the most common primary bone tumor in adults and pediatric patients. There are about 1,000 cases annually in the U.S., so it's a pretty rare disease overall. And the survival, if it's localized, the diagnosis is fairly good, but if it's a relapse, it drops down quite significantly. Unfortunately, most patients actually have micrometastasis at diagnosis, so if they don't get very intensive chemotherapy, almost all of them will develop metastasis. And one of the things that motivated us to focus on this disease is that we haven't really seen any new treatments for over 40 years. And this is just an X-ray that we've seen. Those of us who focus on these tumors have seen way too many times, which is this kind of destructive lesion in a long bone. It's kind of the typical presentation. And we also don't have, despite learning a lot about the biology of these diseases, we still now, as oncologists, we can't really tell which patients are going to do well from the chemotherapy and which patients are going to not respond to the chemotherapy. And that's also kind of a strong impulse for the research that we've done. And there's been many efforts over the last few years to try to improve the outcome for these patients. And this is a very large and very disappointing study that was done in collaboration between labs, groups in the United States, as well as groups in Europe. And essentially what they showed is that what they tried to do was intensify the treatment for patients that they deemed to be likely to have a worse prognosis, and even intensification of treatment by adding two chemotherapeutics called ifosfamide and etoposide didn't really change anything. You can see that these survival curves are overlapping. So that really, to me, argues that we need to learn more about the biology of the disease. We can't just keep throwing more chemotherapy at these tumors if we hope to see a difference. And really what we're trying to prevent is what you see on this CT scan. So this is actually a patient of mine who unfortunately is deceased. And on the left side here, you can see the lung windows on a CT scan, and it looks clear. And then this is after she finished her initial chemotherapy. And then a year later, she came back with this large nodule here, which is a metastatic focus that grew after she finished chemotherapy. And then she went on to develop many more of these and eventually to die of this disease. And this is what we usually see. These patients die of metastatic progression. So at UCSF, we've had a very strong effort over the last 5 or 6 years to collect patient samples from patients at both at diagnosis, but more importantly at relapse, to really try to learn about the metastatic process and learn what is it that makes some of these tumors difficult to treat. So this is just we call a circles plot that shows the colors. It's not so important, it just indicates the different types of tumors that we've sequenced and samples where we've been able to collect multiple samples from the same patients. Overall, our goal has been to utilize a comprehensive omic approach using both analysis of the DNA by using whole genome sequencing and the RNA by using RNA sequencing, again, to understand what is it that makes these tumors progress. But we've analyzed a large number of patients, samples from metastatic patients, as well as samples from patients that have already been treated with chemotherapy to try to understand how they become resistant. And the kind of general workflow that we use when we collect these samples, we collect them, we analyze both the tumor and the normal genome, and analyzing the normal genome is very important in order to be able to identify the differences that are unique to that patient's tumor. And then we use this, again, comprehensive approach where we compare the DNA to the RNA and then integrate the results from the DNA and RNA analysis to try to gain insight into what's happening with these tumors as they progress. And this is just kind of another view of the same kind of protocol that we use. Again, we have a patient who has a primary tumor, in this case, an osteosarcoma, and then that patient can also sometimes develop metastasis. We try to collect samples at the biopsy, so before chemotherapy, at resection, and then metastasis, and then do the whole genome RNA sequencing that I mentioned. And then the other aspect of this is because these are rare tumors, we're trying to expand the material that's available to study. So we implant these tumors into mice. So these are called patient-derived xenografts that grow in mice. And then once they grow, we can extract them and also do whole genome sequencing and make cell lines from these tumors and also analyze those as well. So this gives us material to work with, and much of what I'm going to tell you today is from utilizing these patient-derived xenograft cell lines that we've made in the lab. So this is just another circles plot that shows specifically for osteosarcoma all the PDX models that we've made and where they came from in terms of whether they came from a biopsy, a resection, a metastasis. And again, these decorations here are not so important, but just an indication of kind of what analysis has been done on each one of these samples. We've developed about 27 models for osteosarcoma. We've made about 11 cell lines, and all of these have been extensively characterized using this whole genome RNA sequencing approach. So when we first made this collection of cell lines, we were curious as to how well the cell lines would recapitulate the disease. So when you grow cells in a plastic dish, you always worry that they're becoming quite different than the disease that you're trying to study in the patient. So when we made this collection of cell lines with the characteristics that you see here, this just shows you where we got the samples from, the age of the patient, what the source of the sample was, whether it was a resection or metastasis, and then some of the genomic features. So one of the things I'll tell you about in a few slides is one of the interesting things about osteosarcoma is that every patient is kind of unique and has a unique set of driver oncogenes. And then so we did the RNA-seq analysis, and then just something called a dendrogram, where you're just basically looking at how similar the RNA-seq profiles are from each other, and you can zoom in on that. And what was encouraging to us is that, I don't know how well you can see it from the back of the room, but the patient sample, the PDX and the cell line from the same patient clustered together. So that was actually really encouraging, because we were worried when we first did this that maybe all the cell lines would cluster separately from the patients, indicating that they had sort of evolved in a very different direction. And the other thing that was interesting, and I think it speaks to the importance of doing this kind of very intensive collection of samples over many years, is that there are established cell lines that you can buy from a company called ATCC, which sells cell lines to anybody who wants to pay for them. And there are a few osteosarcoma cell lines in that bank, but those cell lines, when we profile them, they profile very separately. They kind of have their own expression profile, suggesting that they maybe have evolved to be different from the patient samples. Although we do see, even in our samples that we've analyzed, some cases where we see quite a bit of evolution. So this is a complicated plot, and you may not be able to see from far away, but basically what it shows is the chromosomes here, and then the copy number for these individual tumors. So this is the tumor that was extracted directly from a patient, and then the cell line that was made from that same patient. So that's why it has the same number. And what we're doing here is comparing both the minor and the major allele, the two chromosomes for this patient. And because we have the germline, we can do that kind of comparison. And then what this shows here is the major and minor allele. What you can see is that there's just a huge amount of genomic rearrangement. So a lot of chromosomal gains, a lot of chromosomal losses in the patient tumor. And if you compare it to the cell line, similar to the data I showed you in the prior slide from RNA, these are quite similar to each other. So you can tell pretty easily that this cell line came from this patient, right? Because the peaks and the valleys are almost identical. There are some differences which are indicated here. So these tick marks here and major allele, and these tick marks over here indicate where there are differences between the tumor and the cell line. But for the most part, that's a pretty, fairly minor differences. Here's another example though, where there's been a very dramatic change. So this is the tumor and this is the cell line. And you can see here just, by the number of the amount of color that you see both for the major and the minor allele, there's a lot more changes that are occurring, indicating that this cell line maybe isn't as closely related to the patient as we would have liked it to be. And we can kind of quantify that using something called the Jacquard index, which is basically just a measure of overlap between the tumor and the cell line. And so here, I don't know if you can see it very well, but for each one of these comparisons that we've done, most of them have a pretty high correlation, but there are a couple of the ones that are shown in the red arrows that have very distinct, very accumulation of differences. So here you can see that the difference, this is the tumor, major allele and minor allele, and the PDX tumor major allele. So you can see that there's a big difference between the cell line and the tumor. So there's only 0.1%, 0.11% overlap in the Jacquard index, indicating that something happened between the tumor and the PDX, so that between the tumor and the cell line, that the cell line is now very different. Whereas this one here, the change actually happened later because the tumor and the PDX are similar to each other, but they're both very different from the cell line. So there's some evolution going on there. So another way to look at that is to focus on the actual genetic drivers in each one of these samples. So these are actually now the paired samples or the trios where we have the tumor, the PDX, and the cell line. And when you look at it this way, and you look at this sort of what we would consider to be the likely oncogenic drivers for each sample, you can see that they're generally pretty similar. So this particular trio, for example, is driven by these two genes here that are amplified. This one here has all three samples have this amplification here. The ones that are very different, like the cell line that I showed you before, have undergone a process called whole genome duplication that probably happened very early on in the evolution of this tumor from the patient to the PDX. And we know that this kind of whole genome duplication also happens in patients when they evolve towards a more metastatic phenotype. So that might be driving some of the heterogeneity that you saw before. But overall, when you look at the cancer drivers, we do see that there's quite a bit of similarity. And some of the, just to kind of give you a different view of these tumors and a different appreciation for why they're so complex. So this is something called sky analysis, where you can see the actual individual chromosomes that are colored using this sky technology, so that you can see all the yellow is all the chromosome 1s, the dark red is chromosome 2s. And normally, obviously, you should have 2 of each chromosome, but this genome of this osteosarcoma has 7 or 8 copies of chromosome 1, 5 or 6 copies of chromosome 2. This is what we call aneuploidy. So just a massive amount of rearrangement and missegregation of chromosomes, leading to these very abnormal patterns. But the other thing that's quite interesting about this particular case is that what we think is the oncogenic driver in this particular tumor, which is a gene called MYC, which is normally on chromosome 8. You can't probably see it here because the letters are too small, but it turns out that if you label this sample with a probe for the MYC oncogene, you see a MYC signal in almost every chromosome, indicating that when this chromosome shattered and then rearranged, the MYC oncogene kind of got spread all over the genome. And it shows up in what these structures that we call homologously stained regions that are highlighted here in kind of the zoom in, where you see that there's multiple copies of MYC and another oncogene called cyclin E in this particular chromosome, but that's actually scattered throughout the whole genome. And you can see another kind of example of how complex these genomes are. This is a collaboration with a group at Stanford, Christina Curtis's group, who does kind of deep computational analysis of cancer genomes. And what this is showing you is structural rearrangements that are occurring between chromosomes 8, 10, 11, and 19. Each one of these arcs is a evidence based on the whole genome that these chromosomes are coming together. And you can see that signal in the whole genome data. So again, these genomes rearrange in a very complex way, and probably leads to the reorganization of the regulatory regions of the genome that allow these oncogenes to be expressed at very high levels and contribute to the oncogenic phenotype. But despite that complexity, when we start looking across many of these genomes, we do begin to see that there are particular oncogenes, for example, MYC, as I mentioned before, but also other ones like CDK4 and cyclin E that are present not in all patients, but in significant numbers of patients, so that you have 20 or 30% of patients that have mutations in a variety of these different oncogenes. These are the gains, these are the deletions. So we frequently see deletions in RB and P53. So there is some kind of order to the chaos, but this is one of the more complex pediatric cancers that we know of, and definitely a major challenge in terms of trying to understand what's happening here. So much of the effort in the lab has been trying to make order out of this chaos and help us figure out, can we find ways to think about these tumors, maybe not as one disease, but maybe subtypes of disease that could be amenable to treatment with specific targeted therapy. So this is a paper that we published a few years ago now, where we again took the whole genome data, looked for the commonly amplified oncogenes, the commonly deleted tumor suppressors, and then you can see, each one of these columns is an individual patient, and so you can from the back of the room, you can tell that each of the patients is unique in terms of its combination of oncogenes, but you can begin to put them into buckets. Basically, you can think of like a bucket of cases where there's an amplification of cyclin E, a bucket of cases where there's amplification of MYC or amplification of CDK4. And a lot of those oncogenes actually have drugs that have been shown to influence the development of these target these pathways. So what we essentially did was try to simplify this very complex landscape by putting patients into buckets based on the most highly amplified oncogene that would fit into one of these groups, and then treat them with a drug that would match. So, for example, this MYC amplified group we treated with a drug called CDK9 inhibitor, which blocks activation of CDK9 and is involved and has been shown by other groups to be potentially important, particularly required for patients that are driven or samples that are driven by amplification of MYC. So here you can see that by Western blot that MYC is highly expressed in these two PDXs that fall into this group. When we treat them with a CDK9 inhibitor, we get loss of MYC, and we also have data showing that we get loss of several downstream targets of MYC. And then when we do those experiments in mice using the PDX models that I told you about, we can see a very significant decrease in the tumor growth in the MYC treated, the CDK9 treated MYC amplified PDXs, and we also have data showing that the non-PDX amplified, non-MYC amplified PDXs don't respond to this drug. So this kind of begins to suggest that maybe we could find a way to bring precision medicine to the treatment of these patients in a way that we haven't been able to do before. And so I'm not going to spend too much time talking about that because this part of the work is published. We've also been putting a lot of effort into developing models. As I told you at the beginning, what these patients usually die of is not the primary disease, but actually the development of metastasis in distant organs. So in order to develop models that allow us to study that, we think we can take these PDX-derived cell lines and plant them back into a mouse in what we call an orthotopic site, which is basically the hind limb. And then when that tumor grows, we can amputate the leg. The mice will continue to run around, and then we can follow the development of metastasis either by bioluminescence or by micro-CT, and then use the same therapies that I was showing you in the prior slide to see how well these affect the development of or inhibit the development of metastasis. And I think I'm going to skip this slide for lack of time, because what I really wanted to spend most of the hour focusing on is sort of another approach to studying osteosarcoma that is really focusing on the epigenome rather than the genetic complexity. And this is work that was carried out by Eunice Lopez Fuentes, who's an extremely talented a postdoc in the lab, and came to my lab from Mexico and had a background in yeast genetics and epigenetics, and really wanted to have that be the focus of her work in my lab. So she's been a fantastic leader in bringing epigenetic analysis to the lab, and really all the work I'm going to tell you about is hers. And so for those of you who are not familiar with this technology called the TAC-seq, what this allows you to do is to focus on the areas of the genome that are really active in terms of their of transcription. And this works by this technology that was developed by a couple of groups at Stanford, where you can basically use the ability of transposons to integrate into the genome at regions of open chromatin, and then you can use that as a way to basically label the open chromatin and then sequence those regions. So this is kind of the protocol for ATAC-seq. You isolate the DNA, you mix it with the transposase that has these adapters that then get integrated into the genome. You can then amplify those regions by PCR, amplify them, and then you get pileups of the reads into the open chromatin, and then that way you can define what is open chromatin and what is closed chromatin, and it give you a sense of sort of the cell state of that particular sample DNA. So we were interested, or Anissa was interested in using this technology to ask, we've as I showed you before, osteosarcoma is incredibly complex. We may be able to make some headway in terms of using that genomic complexity for targeted therapy, as I showed you, but perhaps the epigenome will allow us kind of a different window into understanding what is happening with these tumors, and can we find ways to attack them that way. And so the first thing that Anissa did was to profile the cell lines I told you about. So we had a collection of about 11 PDX-derived cell lines, as well as some of these commercially available ATCC cell lines. And she did the ATAC-seq analysis, and then she analyzed the data using a principal component analysis, which is basically just a way to reduce this, very complex genome-wide analysis into two vectors, the first principal component and the second principal component. And so what you can see here is that these samples are clustering in two very distinct regions of this principal component of this map. And a lot of the commercially available cell lines are clustering over here, except for this one, HE109, those are shown in pink. But our PDX-derived cell lines kind of are kind of distributed across this this map. And so when then Unisa went on to do this with the PDXs themselves. So these are the tumors that we took out of the mice directly. And again, you can see that there's quite a bit of distribution of these samples in the PCA plot. And you begin to perhaps see the emergence of a third cluster. So you have one here, one down here, and one over here. Then we also, of course, wanted to see, well, what about the actual patient samples? We don't have that many of those, so we haven't been able to do this as extensively as we would like. But we do see, again, some of the patient samples clustering up here, some cluster down here, and one of them so far has clustered on the right side. And the last thing that Anissa did was to then ask, well, does this correlate at all with the developmental origin of these tumors, or does this is perhaps reflected in the attack profile of skeletal progenitors? So we didn't have access to a ton of data from skeletal progenitors, but there was one paper that was published by a colleague at Stanford, Chuck Chen, who had isolated some very early skeletal progenitors from human fetal tissue, and that data was public. And then there was also some ATAC-seq data from a cell line called HFOB, which is a kind of more committed osteoblastic precursor. And what we saw, and what we also thought was interesting, was that these two samples clustered, in very different parts of this PCA. So what Anissa did next was to basically somewhat arbitrarily, define two clusters, what we call cluster one on the left and cluster one on the right. And then all of the analysis that I'm going to show you now was done kind of with this somewhat arbitrary classification. So if you look at the ATAC-seq peaks for cluster one and cluster two, they're very distinct. So these are the cluster one peaks and the cluster two peaks. And you can't see this from far away, but basically this is all this stuff up here is basically just decoration with a lot of the clinical phenotypes that we know exist in these samples, because all these samples are samples that we obtained from Benioff Children's Hospital. So we know the clinical course of these patients, whether they metastasize, whether they have MYC amplification. And so, for example, most of the cases of MYC amplification fall into this cluster, whereas the non-MYC amplified ones all fall into this other cluster. one of the things you can do with a TAC-seq, which is really quite interesting and quite exciting, is you can look at the regions underneath those pileups to look for enrichment of transcription factor binding sites, which would begin to then tell you, why do these different states exist? Why are there some samples that have this particular set of peaks that are amplified, or that are enriched, and another set of samples that have a different set of peaks? And so when Eunice did this analysis, this is where things started to get really interesting. You begin to see that all the cluster 1 samples have enrichment for open chromatin that has transcription factor binding sites that are very involved in bone development. So things like RUNX2, RUNX3, Hox genes, things like that. Whereas the samples in cluster 2 have a very different set of transcription factors that are involved in what's called the AP1 cluster, which is related to RAS signaling, and kind of a completely different pathway for oncogenic development. And Eunice has been able to validate this very nicely using Western blots for these transcription factors. So for example, all of the samples, or most of the samples that have high MYC amplification also have high MYC protein, and they all fall into cluster 1, whereas the samples that have that fall into cluster 2 have high levels of expression of these AP1 transcription factors like FOSL1 and FOSL2. And that correlates also with upregulation of the ERK pathway, which is upstream of AP1. She's also done this at the level of what is actually bound to the chromatin. So this is the total cell lysate, but if you actually focus in on the nuclear fraction and whether these transcription factors are actually binding to the chromatin, you can again see a very strong distinction between the cluster 1 and the cluster 2 samples in terms of their what transcription factors are bound to the chromatin. So all of this is very reminiscent, actually, of work that's been going on for several years now, some of it being done here across the street, looking at another pediatric cancer called neuroblastoma, which is very different from a biological point of view from osteosarcoma, but also happens to have these two cell states, one that's been described as the adrenergic cell state, and another state that's been called the mesenchymal cell state, that also have very distinct transcription factor drivers, as shown in this slide from a paper published a few years ago. But this kind of work had not been done in osteosarcoma. And just to drive this a little bit further, Eunice has also been able to validate that some of the markers that she's seen as differentially expressed in these two clusters, for example, here SADB2 and SP7 are two genes that she identified as being highly expressed in cluster 1. You can see by looking at the immunohistochemistry of the actual tumor samples that there's a very large difference in the positivity of samples that fall into cluster 1 versus cluster 2 in terms of expression of both SADB2 and SP7. And conversely, with the samples that came from cluster 2, they show upregulation of FOSL2. So we think that this is potentially very exciting because it begins to give us a handle on how to think about this disease as not one disease, but maybe distinct diseases that could be amenable to therapy with different approaches. So the next thing that Eunice then wanted to do was to ask, do we see evidence for these two cell states or disease subtypes in patients themselves? And because we didn't, as I showed you before, we don't have a lot of ATAC-seq data directly from samples, we had to kind of go about this in a somewhat roundabout way. So what Eunice did was to first define the peaks for the two clusters, cluster 1 and cluster 2. So this is from the ATAC data. And then using a computational approach called GREAT, you can then assign the peak to its likely most nearest gene that is likely the gene that is driven by that peak of open chromatin, or where the transcription factors are enriched. And so she was able to define a set of genes that are enriched for likely enriched or poised to be expressed in cluster 1, and genes that are poised to be expressed in cluster 2. And it's important to remember that ATAC tells you what could happen, but it doesn't tell you what's happening at that particular moment, because you're looking at these cell lines. And so it's possible that those genes are poised to be expressed under the right conditions, but they're not actually expressed at that moment. On the converse side, you can also take the RNA-seq data itself, which we also had for all of these cell lines. So this is for the cell lines in the PDXs, doing an unsupervised clustering. of the RNA-seq data. We already knew what cluster they fell into because we already had the attack data, but you can see that they do separate even in the expression space to some extent, not cleanly, but most of the samples that are in cluster 1 over here and the samples in cluster 2 over here. And you can see that reflected in the heat map as well. So UNISA also generated a differential expression list of genes from the RNA-seq data, and then basically overlap these two. So the attack genes that are unique for the clusters and the differential expressed genes that are unique for the clusters, and then basically took the overlap between those two. So as I said, it's a little bit of a roundabout way to get at this so that we could get a gene signature that we thought would potentially be mirrored in actual human data, right? So we have quite a bit of human data directly from patients in the lab. So then Anissa took that list of 300 and something genes, 343 genes, and asked whether that data was capable of separating the human samples into clusters. And what she sees, and this is, I think, preliminary, but very encouraging, is that indeed there are two clusters. You can see that in the dendrogram at the top. So there's this cluster on the left, cluster on the right. It's not a super clean separation as what you might expect from something as indirect as I showed you. But if you look, I think you can see without having to squint too hard that these samples on the left have much higher expression of these genes down here, and the samples on the right have higher expression of these genes here, which is what's driving these two clusters. So what we're doing now is to try to correlate this with clinical phenotypes like development of metastasis, development of sorry, let me see. Develop or most importantly prognosis. Does this correlate with the prognosis of these patients? Because if we could show that, then that would obviously have important clinical implications. But speaking of clinical implications, the last part of the talk, I just wanted to mention some work kind of tying this back into the work that I showed you at the beginning with the targeted therapies. So at the same time that Eunice was doing all of this work, another senior scientist in my lab, Leanne Sales, had taken these same cell lines that you see here on the left and treated them with all the drugs that you see at the top here. And this is a list that we came up with in collaboration with my colleague here across the street, Katie Janeway, who's the local expert on osteosarcoma. And these were drugs that she, in her opinion, would be important to test, in a large panel of cell lines like this. So what this shows you essentially is the size of the dot indicates the area under the curve for the dose response, and then the IC50, which is the dose at which 50% of the cells die in this assay, is the color. So what you're looking for is basically the smaller pink dots indicate very strong responses to those individual drugs, and you can see that there are quite a bit of heterogeneity here. So there's some cell lines that respond very well to some drugs, whereas other cell lines don't respond, which is kind of consistent with what I told you at the beginning, that this is a very highly heterogeneous disease. So one day I was driving home and I was thinking, well, why don't we put these two data sets together and ask, are there any drugs that for which the response is correlated with the cluster, these two epigenetic clusters that I told you about. And so this is one of these things, why we love science, because you get results like this, and you're like, oh, this makes it worthwhile, right? So clearly there was some really strong differential responses. In some drugs, it kind of made sense. So if you remember, I told you that cluster 2 is driven by AP1, which is downstream of MEK and ERK. And so it was very reassuring to see that actually these samples that fall into cluster 2 had a much lower IC50 for trametinib, which is an ERK inhibitor. So that completely makes sense. And then a little bit less clear why, but the samples that are in cluster 1 seem to have a much stronger response to these two drugs, one of which inhibits a protein called aurora kinase A, and the other one inhibits a protein called aurora kinase B. So why that is, we're not totally sure yet, but we were pretty encouraged by this kind of correlation between the epigenetics and the drug response. And then Nisa's gone on to validate this with several of these lines. And here's some results with the aurora kinase A inhibitor, where you can see that there's some cell lines that respond very well, and those are the ones that are in cluster 1, whereas the cell lines that are in cluster 2 don't respond as well. And conversely, with the MEK inhibitor, cluster 2 cell lines respond very well, and the cluster 1 cell lines don't respond at all. And that holds up pretty well in vivo. So this is a PDX that we would expect to respond to aurora kinase B, and it responds quite nicely. It does respond a little bit to trametinib, but that's not statistically significant, and the response to aurora kinase B is much stronger. And conversely, here's another cell line where we would expect there to be a response to trametinib, which is indeed what we see, whereas there's no response to aurora kinase B. So we obviously need to expand this to additional cell lines, but I think this to us is again very encouraging. We would, as you probably know, very few solid tumors are actually treated with one drug. So it's not like we're arguing that we found a cure for osteosarcoma, but I think it begins to show us how we might be able to use this information to design more rational combinations that would be specific to specific subtypes of the disease. So the ongoing efforts in the lab right now are to determine the mechanistic basis for these distinct transcription factor activation states. Why is the AP1 cluster activated in one and not RUNX2 and vice versa? Does that have to do, for example, with the developmental origin of the tumor? So it's possible that these two clusters develop from different cells of origin, and that's what's driving the kind of different epigenetic state, something that we still need to understand better. And so those are some of the questions that we're looking at now. So just to summarize, I hope to have shown you and convinced you that the osteosarcoma is a disease characterized by a very highly complex genome, but that nevertheless, using some of these advanced tools, we can begin to make sense of that complexity and perhaps begin to think of ways to apply that to a more rational, precision-driven approach to our patients. And I'd just like to add and to end by thanking the members of the group, which is shown here. And these are the names on the left. And really, most of the work that I showed you about today was done by Eunice Lopez Fuentes and Leanne Sales, but with collaboration from a lot of other members of the lab. And of course, I have lots of collaborators around the world and several here at Dana-Farber. And we've been fortunate to have a lot of support for this from a variety of sources. So thank you very much, and I'd be happy to take any questions. Questions? Don't be shy.
So I'm just wondering, I know you said like osteosarcoma earlier. And you did a lot of profiling. Have anybody done this at all?
Yeah, great question. So we started to do a little bit of that, and we just generated our first single cell RNA-seq data set maybe like a couple of months ago. So we haven't incorporated that into our thinking, but one of the things we're trying to use that single cell data to understand is whether these two clusters are really sort of a yes and no, off and on kind of thing, or is there some heterogeneity at the single cell level, whether maybe some samples on the bulk look more like the cluster 1 and cluster 2, but maybe when you look at the single cell, there's kind of representation of both clusters within each sample. So I think that's going to be important, particularly as we start to think about how to apply this in terms of therapeutics, because obviously if there's, maybe you need to treat both clusters at the same time in an actual sample, right? So that's something that's still ongoing. And we're also using the single cell data to try to understand mechanisms of response to therapy as well, and kind of understand how does tumors respond to therapy. Do they, for example, are they able to switch clusters, right? So if you treat a cell with trametinib, does it then become more like the other cluster, things like that. So yeah, great question, and it's something we're looking at.
So early on, you also showed that patients, the genomic hallmarks of patients is very different from what you actually see in cell lines that you sample. The genome complexity changed to evolve. And so I'm curious as to how does that potentially impact the potential treatment that you know that you're actually changing.
Yeah, so I probably should have described that better. So I think the complexity that we see, the change that we see, we think is mostly driven by whole genome duplication, but the actual oncogenic drivers mostly stay the same. So even though there is this kind of a change in the baseline because everything gets duplicated, and that allows for kind of ongoing evolution. I think that the vulnerabilities probably are still the same in the cell line as they were in the patient, and that's probably what the RNA-seq data is telling us, that the clustering remains the same. But it's a great question, and we have to be careful with that because we don't want to make too many assumptions based on cell line data that then don't translate into patients. So that's why we always try to kind of bring everything back to the analysis of the patient samples. So I was very encouraged to see that we do see some of the the signature that we derive from the cell lines. You can see that in the patients, right? So there may be some ongoing evolution, but hopefully we can capture enough of what really matters to make a difference.
So I'm wondering about the role of AMP kinase inhibitors, whether this could potentially also explain the genomic rearrangements that we have in these things.
You mean like the vulnerability?
Yeah.
Yeah. Well, we should talk about that more. You probably know more about ROR kinase than I do. Obviously it's a gene that's protein that's very important for chromosome segregation, and yeah, I've thought a lot about that a little bit. We haven't figured out how to really understand like why is it. It's not clear to me why that you would think that drug would be important in any tumor, right? So why is there's this differential effect? Maybe it has to do something with the degree of aneuploidy that exists, and so maybe the samples that have a lot more aneuploidy are more dependent on ROR kinase B. So that's something we need to explore a little bit further, but would love to talk to you about it more.
So there is a question on the Q&A of PDT. One is concerning the PDX model, and with their variety of strategies, do they generally mirror what we see in terms of the gene profiling of the metastatic tumor itself? So the PDX.
Yeah. So for the most part, as I showed in that dendrogram with the RNA-seq, we see very close correlation between the patient sample and the cell line or the PDX in at least as compared to the other samples in our data set. So generally, yes, they do correlate very well.
Did you consider using organoid models instead of PDX models? Did you see
yeah, no, that's a great question. We tried many years ago to make cell lines directly from patients. And what we found was that, at least for osteosarcoma, what usually happens is that you just get overgrowth of fibroblasts because tumor cells don't grow very well. Surprisingly, they don't grow as well as you might think. It's actually a big bottleneck to get something to grow. So we kind of gave that up early on because we were frustrated that we were, these are rare tumors. We would go through all this work to get the sample, bring it to the lab, and then we would get nothing in the end. We would just get a bunch of fibroblasts. So when we grew it in the mouse, at least we felt like, well, we could expand that tumor. And then that what grew is an actual tumor and you have some material to work with. That said, there have been people maybe with, better techniques than we had at the time who have successfully developed, I don't know if they would call them organoids, but there are certainly like, early passage samples from patients. So there's a colleague at UCLA, Alice Saragni, who's done quite a bit of work on this, and she has some beautiful data showing that she's able to grow these. And maybe we needed to be more patient, but we thought that the PDXs would be kind of more tractable. And what it has allowed us to do, I will say, is because we've made not only the PDXs, but the PDX-derived cell lines, we've been able to share these with a lot of people. So I think with the cell line panel, we've sent it to at least 10 labs around the world. So that's, at least, sort of, hopefully we'll kind of facilitate work in a disease that otherwise would have limited models to work with. Thanks, everybody, for being here.
So what happened to hospital personnel, we go for the rotation down the road. Can you go ahead and get into the question? If you have other things to do, you can kind of say that with the people that go to other society or.
You want me to close the webinar? Or do you want to leave it open? Just close it.